Laura Hänninen. I'm the clinical instructor at the University of Helsinki, and I would ask our first speaker for this session to come to stage. That would be Lucy. The stage is yours.
Hi, thank you very much for coming. I'm really excited to be here. I'm Lucy Oldham from SIUC. I'm going to present to you some of the results of my PhD on the association between cognitive ability and aggression in pigs. So why do we talk about aggression in commercial pigs? Well, a fighting occurs when unfamiliar pigs are mixed, and this can be very acute and severe. It can also be prolonged with serious welfare consequences. And pigs vary a lot in how much they get involved in aggression, but when groups are mixed, almost every pig will receive some level of injury from aggression. So you might be very tired from meeting lots of unfamiliar people this week, but if we were pigs, this sort of meeting would be an absolute nightmare. I don't know what they think about Zoom. But what we don't know really is about how pigs decide when to fight, when to leave it alone, and how long to fight for and when to stop. We do know that early life experience affects their fighting decisions later in life. There's evidence that pre-weaning socialization can reduce aggression against other unfamiliar pigs. So this has been demonstrated when sows are in farrowing crates and piglets can mix between two adjacent litters. After they're weaned, they suffered less from aggression. They had fewer bites on the middle and rear of their body compared to controls. In addition, in multi-suckling systems where both sows and piglets can mix, the socializing treatment resulted in pigs being less aggressive towards unfamiliar pigs at a later mixing event, and they had a lower duration frequency of aggression and fewer skin lesions. And interestingly, another study combined enrichment and socialization and found that this was effective in reducing aggression in resident intruder tests. Pigs that had been socialized and enriched demonstrated fewer bites, causing fewer lesions on intruders. So we know that socialization is beneficial, but we don't really know the mechanism for this. And what we think and what has been kind of surmised from these previous studies is that they're learning about pigs in general, and this is building their social cognitive skills. And we also know that environmental complexity has a role in allowing pigs to develop cognitive skills. So this has been somewhat investigated, this first level, but what my PhD is investigating is the link between cognitive ability and aggression. What does this performance in cognitive tests actually really mean in the real world for pigs? So just a few definitions or a look at cognition. This has been reviewed in farm animals recently by Naroth et al., but there are some general traits you can measure in animals: categorization, numerical ability, object permanence, reasoning and inferences, tool use. When we look at aggression, we want to know specifically about social cognitive ability. So this is things like discrimination and recognition. So it's just a social form of categorization really and memory. Communication, which is probably a mix of lots of different cognitive skills. Social learning, attributing attention, pro-social behavior and fairness, which are quite complex to test, but probably would be great to reduce aggression if pigs know what it's like to be fair. So we think that the important skills in reducing aggression are being able to tell the difference between unfamiliar pigs, whether they're bigger or smaller than you, how strong they are, and then being able to remember the identity of pigs you've interacted with before and what were the consequences of those interactions. So you need to remember at this conference, if you've met someone before, what did you talk about? It's no good coming up to say, I like your work in rabbits, remembering they work on dogs. That's a nightmare. So here are our methods. We manipulated the early environment so that half the pigs had a socialization treatment. They were in free farrowing pens, and the piglets, but not the pigs, could mix between the two. And also half of the pigs were moved into a post-weaning enriched pens with double the space allowance of the standard pens with deep straw and some toys compared to very sparse bedding. They were kept in their litter groups until the mixing treatment, which I'll describe later on. And how did we test their cognitive ability? We used a range of tests. So we used the social preference test, spatial discrimination test, and then reversal learning. In this short talk, I'll just describe to you the spatial discrimination test. So this is basically based around a box where the pigs can open it with their snout, and inside is either a reward, a food reward, or a punishment, which is a fan turning on and no food. So the pigs were first trained to approach the box in a positive location, which was randomly assigned either left or right. And when they were consistently approaching the positive location, we tested them on a pseudo-random mix of positive and negative trials. We had quite a short time frame. We had 36 trials over 5 sessions. And this was a go or no-go test, which means their responses could either be go and open the box, or no go, don't open the box within 30 seconds. And another way that they could choose to make a no-go response was if they went into the test arena, looked towards the negative box, and then turned around and stood by the gate. We would let them out so they weren't being frustrated at having made a decision, but being kept in for the full time. And a pass meant that they had to get 10 out of 12 consecutive go or no-go choices correct. So then the group mixing treatment is our outcome measure. So how this worked, across every batch, we split them into 2 groups, and we took half of the litter. We only had them in pens of 4 at this point. So we had, this is 1 litter split into 2 and 2 pigs into each pen. We balanced sex and weight across the 2 pens because we know these are important factors in aggression. And we end these 7 batches, so we have a total of 14 pens. My full ethogram, I'm sure you'd love to see it. I'm just going to show you a really small bit that you need to know. I measured biting and fighting continuously. Is reciprocated within 3 seconds. It lasts for at least 1 second. If no further aggression happens within 3 seconds, then the fight ends unless it restarts within 60 seconds. Then that's the same fight. So a broad overview of how they did in the cognitive tests and what happened. So we had for us to be able to evaluate 114 that reached the pass criteria. And we mixed 175 of them. So you can see that we mixed pigs even if they didn't finish the task. We wanted to keep the social structure as I previously described. So here's a brief bit of the results. Overall, in the time that we gave them, 2 out of 3 pigs two trials compared to 25.5 trials for the males. This is still preliminary. Some females performed the correct 82% of trials on average. So what about the results for the regression? We had 807 fights in this first 4 hours over the 14 pens. And as you can see here, as we would expect, most fighting is between unfamiliar pigs. Very few of them were fighting with pigs they previously lived with. Most fights are initiated against females, interestingly. Males against females, about 300 again. And compared to the other combinations involving females. So we expect pigs of both sexes are more involved in fighting with a higher overall duration. But what about the hypothesis? Does a spatial discrimination test predict fighting. And unfortunately, in this preliminary analysis, I've not found a correlation between the two. The cost of fighting and injuries on the body after 24 hours, or biting and fighting delivered. This is not quite the same as I've written in the abstract, because I'd looked at when I individual differences in aggression. But we know that pigs could discriminate according to the sex of their opponent and familiarity. This might not be cognitive, this may be olfactory. And we might kind of initially conclude that different cognitive processes are involved in learning a foraging task compared to social cognition involved in ability. And I still need to analyze the social preference test and reversal learning tests, which test different aspects of cognition and might predict individual differences in aggression.
Thank you. Thank you. Do we have questions on the floor? Here.
Hi, Faith Antoni from Norwegian Meat and Poultry Research Center. I have to admit, I know zero about pigs. I was just wondering, email.
Fight against a female might be the safer option. They're less costly fights. And if you want to fight for dominance, it might be a less of a risky strategy to initiate females.
Okay.
Hi, Roberta D'Alessio, Chagask, and the UCD. Yes.
It follows on
phase, did you measure if there were any differences, how long it took until they learned the no test?
Yes, so the main difference that we found was them wanting to approach the negative. Measure them to and some of, so unfortunately
Hi, everyone, and thanks for the introduction. So I'm Martina. I work with Chagask in Ireland and also the Institute of Genetics and Animal Biotechnology in Poland. So today I'll be discussing the effects of improved gestation housing on sow oral stereotypies and also on the health of their piglets during the suckling period. So just to set the background a little bit, we know that sows face a number of welfare problems, and a lot of those are associated with the suboptimal gestation housing and management that these animals experience. So some of these welfare problems include things like aggression, which leads to skin lesions, injuries, lameness, and behavioral issues are also a welfare problem. So for example, stereotypies. And in this presentation, I'll be focusing in more detail on oral stereotypies. So in sows, this is quite a common behavior, and it results from feed restriction, which is a common practice in sows. So these animals are hungry and they experience a lack of satiety. And in addition to a lack of appropriate material in their environments to satisfy their natural behaviors, oral stereotypies are really exacerbated. So these behaviors, oral stereotypies, they indicate current or previous unsatisfied feeding motivation, but they also indicate chronic stress in sows. And that brings me onto the idea of prenatal stress, which you can see here pictured in the slide. It's a literature review which I've done on prenatal stress if you'd like to find out more. But just in brief, the stress experienced by the sow throughout pregnancy, it can be transferred onto the offspring as they develop in utero. And that can have a number of negative consequences for things like piglet performance and their stress coping abilities later on in life. And it can also lower their resilience and immune function, so it could make them vulnerable to disease challenges. And in this presentation, I'll be focusing on scour, which is a consequence of prenatal stress. And we know that scour is a major welfare problem in piglets. It's also a huge driver of antibiotic usage of in those animals. And that in turn contributes to antimicrobial resistance, which we also all know that it's a big issue at the moment, and many of us are attempting to combat it. So I think that shows that it is important to tackle the problem of scour. And to do that, I think we really need to try and nip it in the bud. So that means trying to prevent prenatal stress, which in turn means trying to reduce the stress in sows. And that in turn relies on improvements to the sow welfare. So how can we improve sow welfare? Well, we know there are systems which are good for sow welfare. So for example, outdoor or even indoor deep straw bedded systems. But in countries like Ireland, this is not possible at the moment. It's also not possible for farmers to, for example, alter their entire building designs or make huge changes in the pens that could improve sow welfare. So in this study, we wanted to test a plausible alternative. So we wanted to make small incremental pen improvements using materials that have already been shown by previous work to improve sow welfare. So for example, using straw in racks which can reduce oral stereotypies. And by making those small additions, we would create an improved housing system, which we would hope would have a cumulative effect on reducing sow chronic stress. And in turn, that could possibly reduce prenatal stress and lead to less scour. So the objective of this study was to investigate the effect of such small incremental pen improvements on welfare indicators such as sow oral stereotypies and also piglet scour. And we hypothesized that the provision of straw and destructible enrichment in the improved housing would lead to lower stress levels in sows. And that in turn could translate into lower oral stereotypies. And in turn, that would translate into lower piglet scouring. So this is actually a part of a bigger study where we measured a range of welfare indicators. But like I said, in this presentation, I'm just focusing on sow oral stereotypies and piglet scour. So just to describe the setup, the farm that we performed the study on was a large commercial farm. And we had 120 sows in 3-time replicates. Half of those were housed in improved pens and the other half in conventional pens. And we mixed those animals into stable groups of 20 unfamiliar animals on day 30 post-service. We also made sure that both parity and sow back fat were equal between the treatments. So in terms of the pen design, both the conventional and the improved housing had full-length individual feed-in stalls, fully slatted concrete floors, and also 2 blocks of wood and 2 chains as enrichment in the group area. And in addition to this, the improved housing also had straw in 3 racks. And you can see in the red circles here where these were positioned in the pen. They also had rubber mats within the feed-in stalls and also in the middle of the pen, as you can see circled in yellow. And finally, they also had a length of manila rope in the feed-in stalls. So those were the small additions that we made in the pens. In terms of the sow oral stereotypies, those were recorded directly using instantaneous scan sampling at 6-minute intervals. And that went on for 3 hours a day, which resulted in 30 scans per day. And you can see in the bubble on the top of the slide, the behaviors that I considered as oral stereotypies. So these were recorded 72 hours post-mixing in mid-pregnancy and then again in late pregnancy. And we had the animals marked with their ID number on the back so that we could observe them from outside of the pen. And then we also calculated the percentage frequency of oral stereotypies per sow per day. The scour in piglets, the presence of that was scored in the farrow in crates, and it was done every 2nd day throughout lactation. So we always aim to start the scoring on the Monday after the farrowing was complete. So that gave us 10 scores per litter. And the table has all of the criteria for scoring outlined, but just to summarize, a score of 0 meant normal dry pelleted feces, and a score of 3, which was the maximum, meant severe diarrhea. And you can see in the photographs for comparison of severity. So those scores were then summed to yield a total scour score per litter throughout the entire lactation. Very quickly, statistical analysis was done in SAS using generalized linear mixed models with PROC mixed to see the effect of the treatment on the welfare indicators. So moving to the results, we saw a clear effect of treatment on both the oral stereotypies and also scour. And I'll show you graphs where it's clearer to see. So this graph shows you the effect of treatment on sow oral stereotypies, and you can see that the sows housed in conventional pens had much higher levels of oral stereotypies than the sows in improved pens, and that was true for both mid-pregnancy and also late pregnancy. And I think it's also interesting to note that although all of the sows in the study entered the study with presumably the same levels of oral stereotypies, the levels increased, the levels of oral stereotypies increased for the conventional pen sows as the pregnancy progressed, and they stayed high, whereas the sows in the improved pens, their stereotypies reduced as pregnancy progressed, and they actually stayed low. So I think that suggests that the improvements that we made were able to not only prevent further oral stereotypies from developing, but they were also able to reduce the stereotypies that were already there at the start of the study. This graph shows you the effect of treatment on scour in piglets. And again, we can see that the piglets born to the sows from the conventional pens had much higher levels of scour as compared to the piglets born to sows in the improved pens. So just as a bit of discussion, it's clear from the results we got that the provision of straw and the destructible enrichment was able to reduce the oral stereotypy levels of the sows in improved pens. And that's likely because it satisfied their behavioral needs and satiety. And this is what we expected because previous work has shown that fiber feeding does have a beneficial impact on oral stereotypies in feed-restricted sows. So we think that the psychological well-being of those sows was improved, and clearly their welfare was improved as well. But we also think that it's possible those sows experienced lower chronic stress levels. And I think further proof for that comes in the form of those lower scour scores in their offspring, which translated into their improved health throughout the suckling period. And so I just want to present, I didn't actually show it in the results section because we got the results after abstract submission, but I think it's worth mentioning that we also saw lower haptoglobin concentration levels in the saliva of the sows in the improved pens. And I think that provides a nice psychological or physiological link between improved sow welfare and offspring health, in addition to that psychological link that I described in terms of the oral stereotypies. So just as a take-home message, I suppose, we did see a clear improvement of welfare to both welfare of the sow and the piglets in the conventional housing as opposed to, or in the improved housing as opposed to what we saw in the conventional housing. And the fact that we are able to lower the scour levels is potentially very, could have potentially very positive implications for antibiotic usage and hopefully for antimicrobial resistance as well. So I think what I want to show here is that even making those small changes is worth, you know, the time, and that farmers should be encouraged to do even that, if nothing else is possible, as it does have a positive effect on not only the sow, but also the piglet. So that's it. And I'd just like to thank everybody who's helped me with the study. And also a big thank you to the ISAE Conference Attendance Fund for making it possible for me to come and present my work. So thank you.
Thank you. Now we have time for questions. And there's, as I check the internet. No, not yet. What about there? Yes, please. And please just tell who you are, where you're coming from.
Hello, I'm Jens Malmqvist from Aarhus University. Thank you for a nice presentation. You attribute some of the positive effect of your enrichment to straw. I have actually two questions. The first one is, you actually use multiple small enrichment. So what made you think straw is the most important? And the second one was about using a straw rack. Some studies say that's not the best way of doing it, but did they use it?
Can you just repeat the first part? Sorry, I didn't
Yes,
catch the start.
you have multiple small improvements, but you only talk about the straw rack or the straw as the mechanism.
Yeah, so, okay, so I'm only talking about the straw racks here really because the focus in this abstract and in this presentation is on oral stereotypies, and we think that the straw had obviously the biggest effect here. But as I said, this is a bit part of a bigger study, and we did measure a range of other welfare measures. So for example, we also scored tear staining around the eyes, which and we did locomotion scoring, which would have related to the rubber floors that we provided. So we do have results on that, and there is positive results there as well. And then the second part was whether the sows used the straw, was it? They did a lot. Yeah, it was definitely whenever I came in to refill the racks, it was the highlight of their day, and it was gone very quickly. So yeah, thank you.
And there was a question over there somewhere, so, yep.
So Megan Hain, I'm from the States. Very interesting study. I think it's sort of really interesting to see, you know, even the improvements that you, as you said, you know, you couldn't do a whole lot, but the improvements that were made, that there was a significant difference. So I was wondering if you had any plans or if you were aware of other studies that were looking at sort of other systems that there was more enrichment in that, and looking at a similar setup to see if there's, you know, more significant changes with a more significant change in the environment.
Yeah, so okay, so this study that I presented here, that's only based on the first 3 replicates of this study, and we already saw significant results here. And on analyzing the full data set on 6 replicates, the results were even better. So I think from my own perspective, yes, it is promising, but I don't actually know of many studies that have done similar things. There are studies that have, for example, used straw racks, but that's just a single edition. So there is not a lot to go on in terms of multiple editions. So hopefully I can build on it, and maybe people can build on it in the future as well. But because I think it's exciting to know that, you know, without having to change a whole lot, like you said, because it's not often possible, this is still good. Yeah. Thank you.
Any more questions? Okay, then we change the speaker. Thank you.
Thank you.
And our next speaker is Agnes.
Thank you. I'm a bit taller. Thank you. Good afternoon, everybody. My name is Agnes Moravčíková. I am a PhD student from Czech Republic, from Czech University of Life Sciences in Prague. And usually I would speak about cows because my dissertation is about cows, but today I chose to speak about rabbits because we have a small project about rabbit welfare, and I thought it would be more interesting for you because this is the only talk about rabbits in this event. So let's get started. So first of all, I just want to say that I will refer to female rabbits as does, and I will refer to their offspring as kids. So just to make sure that you know what does it mean. So usually in a meat industry, those are housed individually in biocages with their kids until weaning. And we are speaking about millions and millions of rabbits because in Europe, every year around 100 millions of rabbits are slaughtered for meat production. But there is this increasing demand for products originating from rabbits reared in nature-like environments, which also means group housing because rabbits are social species. They live in colonies consisted of 2 to 9 females and 1 to 3 males and their kids. So yeah, it's their nature. So that's why, for example, group housing is common in Switzerland. And we also cannot forget about the European Citizens Initiative and the cage age, because it concerns rabbits too. So we should think about some alternatives instead of wire cages. But the problem with group housing is that there is a really high frequency of aggressive events. And it leads to injuries and it can lead to death. But this aggressive behavior is observed also in European wild rabbits, so the ancestor of the domesticated rabbits, where it's really frequent at the beginning of breeding, when those fights for breeding sites and males fight for mates. And then also after parturition, when those can be aggressive towards kids from another mother. So on the one hand, we have this rabbit as a social species, but on the other hand, they could be really aggressive. And several papers strive to solve that. They used different kind of enrichment, such as platforms, straw, dark corridors, but with little or no effect on aggression. What actually had a positive effect was to provide hiding places. And then there is the possible effect of genotype on aggressive behavior, where Morton and his colleagues described that Dutch rabbits were more aggressive than New Zealand rabbits. So we decided to focus on these two findings. And we decided to investigate the effect of housing with the possibility of making burrows, which look something like that, what you can see on the screen, and to investigate the effect of genotypes on aggressive behavior in group house breeding does. So the first hypothesis was that compared to those without the chance to dig burrows, those who got this digging opportunity, they would have less aggressive events, fewer skin injuries, lower kid mortality, and fewer injured kids. And the other one was that compared to commercially used crossbreed Hiplus, those Mecklenburg does from a native slow-growing breed, they would have less aggressive events, fewer skin injuries, lower kid mortality, and fewer injured kids. So altogether, we had 72 rabbit does. They were 24 weeks old. We had 36 this crossbreed Hiplus does, and then 36 this native Mecklenburg breed, which we can find all around in Czech Republic. And we had 2 housing treatments. We had concrete floor with deep litter, so they cannot dig their burrows there. And then we had ground with deep litter, where they had a 2-meter high layer of soil there, where they could do their species-specific behavior. They can create their own burrows. Actually, in both treatments, they also had some nests and elevated platforms. So the setup was the same. The only difference was the soil. And in each group, we had 6 doses. And for one group, we had 3.5 meter per 2 meter space. So during the whole experiment, we observed those 10 times, twice after the group was formed at 10 a.m. and 10 p.m. because they were the most socially active that time. Then after the mail was added, then before kindling, then 2 weeks after the kindling, and then 35 days after kindling before the weaning happened. Each dose was observed for half an hour during these 10 times, and we used 1-0 sampling method with 1-minute intervals to observe aggressive behaviors such as boxing, chasing, attacking, and threatening. And then we also knew the number of skin injuries, and we knew the postnatal kid mortality, and we used generalized linear mixed models to test the effect of genotypes and housing. So if you look at that graph on the y-axis, we have the predicted values of the number of attacks, and the digging group where they could dig their burrows, it's the black color. So the those from this digging group, they had less attacks compared to those from this non-digging group. And it was also reflected in lower number of skin injuries, which you can see here. And this aggressive behavior or aggressive events, they increased with body weight, but among Mecklenburg native does, it appeared less often than among these crossbreed Hiplus does. So on the y-axis, we have predicted values of the number of attacks, and on the x-axis, we have body weight, and the blue color represents the Mecklenburg breed, and the red one belongs to crossbreeds. And over here with the same setup, we found that with an increasing body weight of those, the number of skin injuries increased, and it was more pronounced in this crossbreed Hiplus. And if we look at those kids, we found that housing treatment and genotype had effect on the number of injured kids and also on kid mortality. So over here, the black color represents kids from the digging group, and you can see the number of dead kids, the predicted values of number of dead kids and injuries on the top. And here you can see the effect of genotypes, and the black color represents this crossbreed Hiplus. So we found that housing of rabbit does with possibility of making burrows decreased the aggressive events, and we saw that they didn't use the soil just to dig, but they created their own burrows. They would hide their kids there. Sometimes they would seal the opening of the burrow to make sure that nobody can get in. So they really used the burrow to actually hide there, not just to dig and play with the soil. And it resulted in less injured does and less injured and dead kids. So we really think that it's important to provide hiding places and to think about what kind of hiding places we should provide to those, because they didn't really use some boxes what we gave them. They really wanted to hide in those burrows. And we also found that Mecklenburg rabbits manifested less aggressive events compared to commercially used hybrids. So it could be nice to think about what breeds we could use for these alternative group housing systems. Thank you for your attention. And if you want to discuss some other things about rabbits, I will be around.
Thank you. Do we have questions? Yes. The lady in the back. Yeah.
Okay. Sabine Gebhardt from University of Bern. Thank you very much for this wonderful talk, because I like this approach to the problem of aggression very much. But I was wondering or a bit worried about the health of the rabbits. How did you control coccidiosis, or how did you clean this system. So do you think this could be used in a commercial setting?
Yeah, so actually, I think that this could be used, but it's small organic farms, so it's not for a big setup. And I think we should use some kind of rotation. I don't know if, you know, you can imagine it as we rotate pasture, for example, that we should rotate the pens and to somehow put a new soil there, because you can imagine it like a bath where you fill it with soil and there they can dig there. So I think if you have some kind of rotation and you change the soil, it can maybe help. But it's if you want to do it in a bigger scale, you should more think about how to kind of make this burrow system from something else, what you can actually clean. So now we are thinking about how to create a similar environment from materials, from plastic or something, where they could like hide similarly like in burrows, but it's not from the soil. So you can have a better hygiene. So it's the next step to kind of think about, yeah, that way.
And there were some more hands above. Yeah. There.
Hi, very good that you seem to have found something that actually works, because this is a real challenge.
Yeah.
I was looking at your slide with the lesions, though. It seems that in both groups there were quite a high number of lesions. Did I see that correctly, or?
Let's see. I don't remember the number of the lesions, but for example, if we compare the mortality, the best for if you had the combination of the Mecklenburg breed and the digging, there we had 5% mortality. And when we had the hip loose and the non-digging, there we had 35% of mortality. So yeah, but I mean, still, if you provide those, yeah, it's still a problem if you have them group housed, you still have the aggression there.
Misunderstood one of your slides, but I think one of the previous one gave the line for skin lesions, or not? One with the red and the blue lines.
This one?
The next one, I think. Yeah.
No, it was just a tendency.
Right, but in absolute numbers, is that like in the blue group 75 skin lesions per doe, or?
No, it was in groups. It was counted in groups.
Ah, okay. So how many rabbits are in there?
6 rabbits in one group.
Okay, so but still then 12 lesions per doe, and that's in the lower group. That's still quite high.
12 lesions per group.
Okay, so only 2 per doe then. Okay, right. Then that makes a very different
it makes
number.
sense now. Okay.
Yeah. Okay.
Great.
Thank you.
And lady in red over there.
Thank you. The lady in red is Lisbeth from the ILVO. And I was wondering, I was interested in your breeding differences, your genetic breeds. And I was wondering, since, Gila and He Plus are the most used breeds.
Yes.
Do you think it would be possible to, within those breeds, select for more social does who are more appropriate for group housing?
I don't know. I'm not a genetician, so I cannot really answer that. But yeah, if they ask us how to actually house these does and what breed to choose, we tell them that it's better to choose the breed, not the crossbreed, because it's just not. I mean, they were bred for meat production, and it's somehow the aggression could somehow like be the side effect or something. So yeah, maybe it's good if we go back to the pure breeds and start with that one. But I don't know if yeah.
No, I agree.
Yeah. Okay.
Any more questions? Well, I have a short one, I hope. It's a short answer. You said that in the future you're going to think about how to have artificial burrows.
Yes.
So the thing is not them to dig it themselves, it just have to be like that they like it.
Yeah, because we don't know really if it was because of the digging or because that they would hide there. So we would like to dig in those data more and find out what was the cause. Yeah, and then we can work on it. And yeah.
Well, thank you. And next speaker, please. Our next speaker will be Kate. Stage yours.
Hello? No. Yep. All right, well, hello everyone. My name is Kate. I am from the University of Portsmouth, and I'm going to talk you through today the results of the first study of my PhD, which looked at risk factors for stereotypic behaviour in ungulates. So what I do just to start with is just define what we mean by stereotypic behaviours. So these are repetitive, invariant behaviours, and they're induced by motivational frustration, attempts by an animal to cope with its environment, or by dysfunction of the nervous system. And they're widely reported across captive species, as we've already heard today. And most importantly, to think about and why we want to study them is that they can have significant implications for management and welfare of these animals. So we tend to group stereotypic behaviours into two groups, so oral and then motor or locomotor. And up on the slide there, you can just see some examples of common stereotypic behaviours we see in ungulates. Oral stereotypies being the most common, as we've already kind of seen in the pigs earlier, but also we do see some locomotor stereotypic behaviours. So up on the right there, the top corner, probably the only mention we'll get in this conference, they're from camels. Shout out to camels. So those are just traces around stables. So box walking at the top and then pacing is movement along one long side of the enclosure. Now, we don't know that much about stereotypic behaviours in ungulates beyond these really key species. So for some of the really, common or, well-known species, we know a little bit about the within species risk factors. But as you can see up on the slide there, nearly all of these relate to captive husbandry or housing. And actually for most ungulate species, we know nothing at all. But yet, in the literature, we see vast differences in not only the type of stereotypy across different species, the prevalence and kind of the frequency that they occur. So maybe a little bit ambitiously, I said I'll just fill out the gaps in this kind of data set. So the aim of this project was to identify species level risk factors for stereotypic behaviour across ungulates, and accounting not only for their captive conditions, but actually also thinking about their wild behavioural biology. And we hypothesised that risk factors would fall into three main groups. So firstly, those involving or relating to feeding and foraging. Secondly, factors relating to movement and ranging and kind of enclosure size. And thirdly, maybe factors relating to the disparity between a species' natural social organisation and that they experience in captivity. So how did we do this? Well, I conducted not one, but two systematic literature reviews. Yes, my lockdown was really fun. The first one was to look for data relating to stereotypic behaviours across all ungulate species. And this generated data for 38 species altogether. And then for these 38 species, I conducted a second review, looking for data relating to those species' wild behavioural biology variables. And I'll tell you a little bit more about them on the next slide. Oh, the next but one slide. So this generated a data set of 96 different captive animal studies, encompassing 38 species, and rather amazingly representing over 15,000 individual animals. Now, it's really important to note here that we did not have all the data for all the captive animal studies, and we definitely didn't have wild behavioural biology data for all the different factors we wanted to look for. So we've got a very patchy data set with lots of missing values, and this was something we had to account for when we were analysing. So to analyze this data set, I actually worked in a Bayesian framework. And this was because modeling in this way allowed me to control for phylogenetic relatedness. It allowed me to include random factors. And really importantly, and something that's not been done before in these kind of cross-species analyses, is that it meant I could keep each individual captive animal study in as its own data point, rather than having to take an average for the species, which is what's been done previously. So the factors that we looked at, so those relating to ranging and movement, those relating to captive feeding, and then those relating to social behaviour. Just to note here, the ones in green are to do with the wild behavioural biology of species, and the ones in purple are to do with the captive conditions. So we ran a separate model for each of these, and our output variables were prevalence of stereotypic behaviour within a population. And then also we ran some exploratory models on the proportion of time spent performing oral stereotypy. Because we've not got much time today, I'm just going to focus on the stereotypy prevalence variables, models. But we've just been accepted to publish this in Proceedings B. So if you want to know more, you can find out about it very soon, or you can come and speak to me later today. All right, so of these variables, we found 4 to be predictive of stereotypy prevalence. So what I'm going to do now is I'm just going to talk you through those 4, just for speed. So firstly, relating to feeding, was feeding strategy in the wild. So these are predictive plots from our models. You can see the different feeding strategies along the bottom there, mixed being a mixed strategy of both browsing and grazing. I'm not sure how well you can see them, but on the bottom there, you can see the sample size. NST means the number of studies that were in that group, and NSP means the number of species that were represented by that group. And obviously, because this was kind of data that were pulled from the literature, they're not even, and there was no way of making it so. So you can see here, really interestingly, browsers and mixed strategy feeders have a higher stereotypy prevalence than grazers. And this is really interesting. Browsers have really highly specialized feeding behaviors and foraging behaviors, having to move between food patches much more than grazers. And these needs to carry out browsing behavior are way more difficult to replicate in captivity than grazing. Okay, so we think that the behavioral need to browse is kind of being thwarted in captive conditions, or they're not being able to meet that behavioral need as often, and that's potentially why there's higher stereotypy prevalence in those species. If we continue to think about feeding then, in captivity, feed availability and feed type are both predictive of oral stereotypy. Now this doesn't, of stereotypy prevalence. Now this doesn't come as a huge surprise. I mean, we were just a lot of you were in the pig talk not long ago. And this has already been shown in quite a lot of single species studies that these are predictive of stereotypy. But it's really useful to know that even cross species, this relationship exists. So we can now take those results and kind of apply them to species that haven't really been studied before. And finally, and I think this one's the most interesting, onto results for sociality. So we actually found that mating strategy was predictive of stereotypic behavior. Now, mating in general is often overlooked in terms of animal welfare. And I think it's something that we really need to start thinking about, or at least this indicates that we'd need to. So we found that promiscuous species were more likely to show stereotypic behavior than polygynous species. And if we think about behavioral needs again, promiscuous species probably have their behavioral need to carry out mating behaviors, or relating to mating more frequently than other species, and that's why they're more likely to develop stereotypic behaviors as a result. So just for the last few slides then, I just want to talk about kind of what this means in terms of animal welfare and going forward. So if we think about those high-risk species, those browsers and the promiscuous species, we need to really start thinking about whether they're suitable for captivity, because at the minute, we're not meeting their behavioral needs, or at least not in a lot of collections. And maybe it'd be useful for collections to start thinking about lower risk species to promote kind of good welfare and reduce stereotypic behaviours. But we can very much appreciate that these animals are already in captivity and we need to do something now. So where we do have browsers, we really need to start thinking about how we can target enrichment to encourage those natural browsing behaviours and those movements between food patches. And we really need to start thinking about what we can do in terms of mating programs. You know, perhaps, I don't know, allowing mate choice or more mate choice for these species, or just allowing them to, you know, actually mate sometimes, whatever that may entail. Because mating is something we really highly control in captive populations. Now, those are the high-risk species, but for our lower-risk species, I think we can still think about using diet to improve their welfare by reducing stereotypic behaviours. So really focusing on low concentrate and high forage diets and providing ad lib access to feed wherever we can. Okay, and finally then, our data set, I can totally appreciate, had lots of gaps in it, and we really want to fill them and improve our models going forward. So what would be really useful to all you animal behaviour researchers is when you're recording animal behaviour, if you could start thinking about oral and locomotor stereotypies and recording them separately, that would be amazing. We really want to dig into the difference in the drivers between the two different types, which is something we just couldn't do with the data that was available here. So with that, I'd like to thank kind of all these different zoos that provided me with some data as well, BIATSA for their support, and some lovely students who helped me with screening some of the thousands of journal articles that I had to go through. Thank you for listening, and I look forward to your questions.
Thank you. There's a question right over there.
Ruth Newberry, Norwegian University of Life Sciences. Very nice talk. I had a question about whether you looked at the difference between males and females in prevalence of stereotypies, especially in relation to the promiscuity aspect.
We wanted to, is the short answer. We really wanted to, and we could totally appreciate that actually with mating system, there's probably big differences between males and females, but there just wasn't sex segregated data available in most of the studies. So it just wasn't something we could look at. But again, going forward, if people would like to report that, that'd be really useful, because we'd love to dig into that in the future.
Hi, Jake Rundle-Worthington, We're All Mammals. Thank you for the talk. You've reminded me of some of the things that I do in my work, which are useful. In your conclusions, you say maybe you should start thinking about not having some species in captivity, and then you also say maybe we should let them mate. How do you square that paradox?
I don't, but no, it's about, I'm not necessarily just thinking let them mate, maybe just performing mating behaviors or having some kind of proxy for those behaviors. I totally appreciate that we don't want to increase the number of animals in captivity that are at high risk of reduced welfare. But if you think about livestock, quite often they don't ever get to mate, and we do want them to breed. So, but obviously in terms of wild animals.
Sorry, then, just to follow up on that then, why not present a solution of just improving those captive environments rather than thinking about just preventing the animals from being in the captive environments?
I also suggested ways to improve captive environments, but I'm not suggesting that we don't have any animals in captive environments. What I'm saying is, if particularly zoos and collections wanted to focus on lower risk species, that would be beneficial. But also, if we do have them in captivity, let's improve their environment. So using enrichment, you know, that kind of thing.
Thank you. Now to change the speaker.
Thank you.
Thank you much. And now our last speaker for today's session is Ricard. Stage is yours.
Okay. Hello, everybody. Congratulations to the ISAE Congress for these organizations. And thank you to invite me to present this work that is called Effect of Access to Pasture on Hair Cortisol and Behavior of Sheep. My name is Ricard Pares. I'm a researcher from the Department of Animal and Food Science in the School of Veterinary Science in the University of Barcelona. And the other authors are Paul Young, Xavier Mantec, and Xavi Suc from the same department. And Xavi Suc is also a member of the ruminant research group in the same university. So sheep flocks are usually managed in a very diversity of systems in most of the countries, and particularly in Spain. We have these extensive systems in which there is no grazing restriction because ewes can access to pasture every day. And then in these systems, abnormal behavior rarely appears. The systems are used breed types adapted to climatic conditions. It's important to consider that at least in Spain, we have different climate regions and geographical conditions. And it's an important factor to have a farm with an extensive or intensive system. We have intensive systems, even that they are not widely used, but they also exist in sheep farms. In these farms, there is clearly a grazing restriction, and then abnormal behavior could appear. In these kind of farms, usually are used productive breeds to have a high production. And in the middle, we have these semi-extensive or semi-intensive systems that have variability in grazing restriction. This grazing restriction appear only in specific circumstances. But in these circumstances, abnormal behavior could appear, and then it's important to know these circumstances, and it's important to consider as well that there is a diversity of breed types in this kind of production. So this question in these semi-extensive systems is when appears this grazing restriction? It could appear in some specific moments of the reproductive cycle. Basically during gestation or last weeks of gestation, or even in the post-lambing period. Even if they are kept indoors all day, this is better for the welfare because the farmer can have a better care of the ewes and even of the lambs, and they get lower mortality, for example. But there are other specific adverse situations. For example, lack of grass. For example, in Spain, in some areas and in some periods, there is a lack of grass, or in drought situations. Unfortunately, we have this climate change in which every day increases, unfortunately, the drought situations. In these situations, usually the farmer kept the ewes inside and they are fed in the barns. So taking account these considerations, and in order to improve sheep welfare in flocks, the objective of our study was to analyze this effect of the grazing restriction on sheep welfare. Okay, we used a flock in the experimental farm in the university with 40 pregnant Ripollesa ewes. They were housed in an open barn, and the experimental period was of 10 weeks. The Ripollesa ewes is the Ripollesa breed is one of the breeds in Spain in the northeast. So the main breed in Catalonia, and is well adapted to these semi-extensive or semi-intensive systems. We had two groups, the control groups, 20 ewes, that have access to the pasture 5 hours each day. That's the usual management of this flock in the university. But the other group, the experimental group, was kept indoors throughout the experimental period, throughout the 10 weeks period, in which use were supplemented because in order to avoid differences in body condition in both groups. The first parameter was cortisol level on hair. And during these 10 weeks, 3 samples were taken. The first one was at the beginning just to remove the hair. And after 5 weeks, we take another sample, and at the end of the study, at the week number 10. The sample was taken from the left forelimb of the lambs. And the other parameter we studied was behavior. To study behavior, it was a video recorded during the 10 weeks, 2 days each week. And each of the days, we have these 3 periods of video recording. In the morning, 15 minutes at 6 a.m., in the afternoon at 4 p.m., 15 minutes, and in the evening at 9, 15 minutes. So in total, was 30 hours of video recording, 15 for each group. And in the video recording, what the ethogram includes was these 4 behaviors. The first one is the aggressive behaviors, headbutting and threatening by continuous sampling. This is a kind of behavior that you can see usually in one farm, in a normal visit in one farm. The second one is an abnormal behavior specific of the sheep, that is wool pulling by continuous sampling, what is studied. And in contrast, it's difficult to see in farm this behavior. The third one was resting of the flock by scan sampling and rumination by focal sampling. Regarding the results, hair cortisol, you can see here that after 5 weeks, the levels were not different between the both groups, the experimental and the control group. And 5 weeks later, at the end of the study, the hair cortisol levels were higher, but were higher in both groups. So there is no effect, at least with these results, of the experimental group in terms of hair cortisol levels. But when we were analyzing all the data, we saw that there were significant differences depending on the use carrying 1 or 2 lambs. You can see here that after 5 weeks, there were no differences, but at the end of the second sample, use carrying 2 lambs have higher levels of hair cortisol. Regarding the behaviors, in aggressive behaviors, you can see here in the red bars, the number of aggressive behavior in the experimental group that you can see that were in higher prevalence than the control group in these blue bars. So, and it was seen in practically in all weeks. So these differences were significant. As I told you, this is a behavior that it is easy to see in the in farms. We have a short video in which in a few seconds you can see this behavior. This is an example when you mix, for example, unfamiliar animals that when they get together, this behavior is usually sometimes to see in a farm. So it was one of the behaviors that increased in the experimental group. The other one was wool pulling, and here you can see again in red bars that wool pulling was not seen in the control group in the 10 weeks. And even if in a very low prevalence, but it was seen in the experimental group. But it was seen after 5 weeks of this grazing restriction. And here you can see again, in just a few seconds, this behavior in which you can see one of the ewes of pulling the wool of the other ewes. And here you can see how the use is pulling the wool. That's interesting because we saw it and we recorded it, but you usually don't see it in farms. Only in extreme situation, if you have a very intensive farm during all weeks kept indoors use, maybe you can see that, or maybe you can see the wool with differences. And regarding the last parameters, resting and rumination, we didn't found, sorry, okay, differences in between two groups. So these simple but interesting results are these three ones. First one is that grazing restriction causes wool pulling in the flock, but it appears after 5 weeks of the grazing restriction. The second one is that you can see always as well an increased aggression, but it doesn't affect the grazing restriction, the resting and rumination. And the third one, that grazing restriction in our study has no effect on hair cortisol levels. That's all. Thank you for your attention.
Thank you. Questions? Over there, go first.
Hello. Yeah, okay. Roxanne from Switzerland. Thank you very much for a sheep talk. There's not so much here. How were the space allowance on the pasture compared to the indoor group? So was it the increased aggression and headbutting maybe because they were like on a more confined space than on the pasture because they just couldn't go like around each other and then they started it? Or?
No, they really were outside with men and 5 hours. So the difference was really so different. It was not the case just surrounding at the farm. They would get out, walking maybe some kilometers even. So the difference was, I think that the most important thing is that it was for 5 hours each day. It's not too long, but 5 hours comparing to no going outside is important.
Hi, thank you. I'm Michelle Reeves from SRUC. I was wondering, since you saw an effect of time on the wool pulling, was there any effect of time on the aggressive behavior? Did it get worse or better with time?
Which time? Yeah, there were some significances. These are only the main results. But in the evening, for example, you never see wool pulling or aggressive behaviors. It's in the morning or in the afternoon. This is another important factor, but because of the low prevalence of these behaviors, I didn't show the results. But I think in other studies, it could be an interesting factor to analyze, for sure.
And over there, lady in green.
Hi, Catherine from SIC. I found your talk really interesting, but to adapt your study to other countries where animals are also kept in, sheep keep inside for months during the winter, what are your next steps in your study? Are you looking at different sizes of barns and so on?
Yeah, that's an interesting question. I was thinking about that in the case of Spain. I think that it's difficult to think about the practical consequences, but anyway, I think it's important this period of 5 weeks, because if you close the use just for lambing, for example, on a specific moment, it's not a problem, probably, it seems, in terms of animal welfare. But in other cases, some farmers say, okay, I have a problem, so the solution is to feed the ewes in the stable, and that's wrong. But when for climatic conditions or whatever, they can go out of pasture, it's difficult to have an answer.
Any more questions over there?
Thank you. Suzanne Millman from Iowa State University. And as a former shepherd, I can also attest that wool pulling can be quite a problem, but it's a relatively rare behavior to see. So I wondered if it was possible to do indirect measures such as fleece score, or if any of the fleece shows up in the droppings.
Yeah, that's a very interesting question, because in another job, we proposed a score for wool pulling that we presented last June in Edinburgh. It was a poster. This is really very important, and that's the question, because if you can see, if you have a welfare assessment on farm, it's very difficult, even if you are 30 minutes to see this behavior. But within a score from 0 to 3, analyzing the quality of the wool, it's absolutely sure that you can measure this behavior. In fact, we saw that in the intensive farms, this punctuation was always up to 1 or 2. In the extensive was always 0. So the question is that the behavior is underestimated. The behavior is not easy to see, but you can score this behavior.
Okay, thank you, Ricard, and thank you for the speakers for this session. Now it's time for coffee, and after that is AGM for the ISA members. Please don't forget it. Thank you.